All right. So I'm going to go over this enzymatic and cellular reactions activity. If you click here, there is a video talking about lactose being broken down in infants. Then this right here is just going to talk about a little bit about proteins. Then right here, we're going to talk about the different types of proteins. We have enzymes, which carry out chemical reactions. We have messenger proteins, which are like hormones that transmit signals throughout your body. We have structural components. They do what they say, they provide support. These are largely found in muscle tissue. Then we have transport and storage. So this is going to carry small molecules within your body. So over here, some of our key terminology that we need to be familiar with is catalyst. So catalyst is going to be something that helps a chemical reaction occur, and it's going to specifically decrease the amount of activation energy needed for that reaction to occur. So, for example, that's like someone who drinks coffee in the morning. That's the energy they need to get their day started. So, without a catalyst, it would require a lot more energy for reactions to take place. And if that continuously happened, then your body wouldn't have energy to do other things. So, enzymes allow your body to do a lot of its activities with a lot less energy requirements. Then we have a model. So, we're going to identify the different parts. So we have the first thing we need to look at is lactose. Lactose is going to be the molecule in milk that you are going to ingest. It's also going to be called the substrate. So it's going to be this purple thing right here. It's going to be the lactose. It's what's going to be broken down. So you would type the word lactose here. Lactase, anytime you see something that says ASE at the end, that's going to be the enzyme. So that's going to be this blue right here. This active site is this area right here. So the active site is the shape of the enzyme where it is going to join with the substrate. They have to be the same shape or it will not connect. This right here is the enzyme substrate complex. It's when the substrate fits perfectly into the active site of the enzyme. We call that lock and key. And then this is going to be the products. So the lactase is going to take the lactose, it's going to fit it into its active site, and it's going to break that down into glucose and galactose. So right here, we're going to be putting the reactants and the products. So the reactants is what's going to be going into the reaction. So that's going to be over here. The lactose is what goes in. And then glucose and galactose come out. So the lactase is not a reactant or a product. It is simply where the reaction takes place. So that at the end of that reaction, that enzyme is going to just sit there and it's going to wait for another lactose molecule to come. So we don't want to use energy making more and more and more enzyme molecules when that one can just be reused. And then right here, if we look, a reaction without an enzyme is going to require a lot of energy. And one with an enzyme is going to require a lot less energy to do the same amount of work. So one thing about enzymes is whenever the pH changes, so say eat something or medication, it causes the pH to change in your bloodstream, that can cause denaturing, which is the shape. It's changing the shape of the protein, so they're no longer going to fit that active site anymore. This also happens with when they're exposed to high temperatures, like a high fever for extended period of time. This active site right here is going to warp, and so then it no longer fits that shape.